Hello everyone, my name is Anushka Dubey, pursuing MSc Biotechnology from School of Biotechnology, DAVV Indore. So today I'm going to present on topic TI plasmid and its subdivisions like tDNA, RI plasmids and disarming TI plasmid. So let's start. So topics to be covered is TI plasmid, tDNA, disarming TI plasmid and RI plasmid. So let's start with the topic, that is general introduction. So TI plasmid or tumor inducing plasmid is an extra chromosomal genetic material found in Agrobacterium tumefaciens which cause crown gall disease in certain dicot plant species. TI plasmid is naturally occurring plasmid and it is a Agrobacterium is a gram negative bacteria. So here I have shown picture of crown gall disease. This is how the formation of uh, nodules like tumor formation in plant can occur, as I've shown in the pictures. And cartoon of agrobacterium bacteria is there, in which chromosome and its extra chromosomal material, that is TI plasmid, is shown. So it's a general introduction that this is how the disease looks like in plant. So, TI plasmid, tumor inducing plasmid, is a large size, around 200 kilo base pair in length, and it has several regions. Mainly, three regions are very important one, that tDNA region, Vir region, and opine catabolism region, along with origin of replication is present. So, let's start with the tDNA. So it consists of right border and left border sequences which carry genes responsible for inducing tumor. Foreign gene of interest can be inserted in this region. So it means that जो tDNA का region है वो लेफ्ट बॉर्डर सीक्वेंस और राइट बॉर्डर सीक्वेंस जो बहुत इंपॉर्टेंट पार्ट है जो कि इन्वॉल्व होते हैं टीआई प्लाज्मिड जब tDNA को इंसर्ट करता है या ट्रांसफ़र करवाता है प्लांट क्रोमोसोम में, तब जो ये लेफ्ट बॉर्डर और राइट बॉर्डर सीक्वेंसेस हैं ये बहुत इंपॉर्टेंट रोल प्ले करते हैं और यहीं से कट होकर tDNA का जो पार्ट है वो ट्रांसफ़र होता है प्लांट्स में. और इस tDNA के अंदर जो ऑक्सिन, साइटोकाइनिन, ओपाइन रीजन है, ये जो रीजन्स हैं ये भी नेचुरल जो टीआई प्लाज्मिड होता है, नेचुरली जो उसकी फंक्शनिंग होती है वो ये होती है कि ये जो तीन पार्ट्स हैं ये रिस्पॉन्सिबल होते हैं प्लांट्स में ट्यूमर कोज करने के लिए. Auxins and cytokinin, both these are the phytohormones which are responsible for the growth production in plants. तो जो ऑक्सिन, साइटोकाइनिन एंड ओपाइन रीजन है ये मेन रीजन्स हैं ट्यूमर कॉज करने के लिए. ओपाइन रीजन जो होता है ये मेनली रिस्पॉन्सिबल है बैक्टीरियल एंड प्लांट की कम्युनिकेशन के लिए. लाइक अगर जो ओपाइंस हैं, ओपाइन सिंथेसिस जब होता है तो the plant gets signalized, or we can say they can cross talk with each other by as the TI plasmid form the opines, so they can cross talk with each other, and this tDNA with the help of virulence region get transferred to the plant chromosome. As I have written that Vir region carries gene responsible for the inducing tumors, responsible for the excision, integration and transfer of tDNA into the plant chromosome. So next is opine catabolism region, that is the region catabolizes opines which are specialized amino acids. As opines are very important as they are the source of energy for the bacteria, as they contain opine catabolism region in their plasmid. And other region include the origin of replication, tDNA border sequence, plant hormone synthesis region and conjugation region. There are basically different types of opine formation occur in the plasmid, or we can say there are different sequences which are responsible for different opine formations. So basically I have taken two examples, that are nopaline and octopine. Nopaline, it has a continuous region of tDNA which is approximately 25 kilo base pair in length, and it produces an opine known as nopaline. And octopine, it is subdivided into two regions which are 13 kb and 8 kb long. It produces an opine known as octopine. It's not that important, but it's just for an information that कि जो TI plasmid होता है उसमें different types of opines produce होते हैं, और उनमें से दो एग्जाम्पल मैंने लिए हैं, वो नोपालाइन और ऑक्टोपाइन के हैं. तो जो नोपालाइन होता है वो 25 kb लेंथ का कंटिन्यूस होता है, लेकिन ऑक्टोपाइन के टू रीजन्स होते हैं. एक 13 kb का होता है, एक 8 kb लॉन्ग होता है. एंड आई हैव शोन दिस थिंग along with the diagram also. It's both are similar, just the difference is that the opine formations are different. Okay, so this is how it's a generally divided into two types of plasmid carrying different opines or forming different opines. Next is the characteristic of DNA. So transfer DNA, tDNA is also called transfer DNA, which is 15 to 30 kilobase pair of the sequence. Opine synthesis is the unique characteristic of tumor cell, as I have already explained this thing कि जो ट्यूमर सेल्स होते हैं वो ओपाइन प्रोडक्शन करते हैं. सम ओपाइंस आर शोइंग हाई प्लांट स्पेसिफिसिटीज. कुछ ओपाइंस जो होते हैं वो स्पेसिफिक प्लांट मतलब हाई स्पेसिफिसिटी शो करते हैं, बाकी इट्स ऑल नॉर्मल. tDNA border are generally 25 base pair, and it's highly conserved directly repeated DNA sequences, that is flanked DNA, tDNA, right and left border. As I have explained ke it's very important part, as because it is the only region, like in whole TI plasmid, tDNA is the only region with which get transferred to the plant or host chromosome for the tumor for the tumor formation. And the reason for the tumor formation are the auxin, cytokine and opine. The genes which form auxin, cytokine and opine, these are the responsible one. So naturally occurring tDNA codes for opine synthesis and phytohormones to induce tumor genesis. Next we have the structure of specifically the tDNA. Here I have shown the oncogenic region. It may be shooty locus or rooty locus. It have the region shooty locus and rooty locus. So oncogenic regions with the gene for auxin, that is TMS1 and TMS2 shown in the diagram, and the cytokine and TMR, which are plant hormone that cause tumor formation, and also contains gene OCS for opine synthesis. Here octopine gene is present. So opine catabolism use bacterium as a food source. Okay. Next, let us see in detail what is rooty locus and shooty locus. So rooty locus is the tumor morphology root, TMR, which produce cytokine. ये जो जीन है, ये जो सीक्वेंस है, ये cytokine produce करता है, and cytokine is responsible for the growth. But also it is root inhibition. ROI region is also there, or we can say it has potential depending upon the plant need. It may produce cytokine to promote the root formation, or we can say root multiplication or growth, more specifically the root growth. But whenever plant need to stop it, it can inhibit the root, like root inhibition also can be occur. Next is the shooty locus, that is tumor morphology shoot, TMS. It produces auxin, which is responsible for the shoot growth of the shoot, but it also contain SHI region, that is shoot inhibition region. So this is the main part of the tDNA, or more specific structure of the tDNA. Let us move to the disarming of TI plasmid. So it is a very important step, or it is a very important we can say process by which we can transform TI plasmid for beneficial use. Like we can use the TI plasmid for the betterment of the, we can say, if we want to transfer any gene of interest like resistance, pest resistance, disease resistant gene, or any kind of antibiotic resistance, jo bhi any beneficial gene, if we want to transfer it to plant, so we can use TI plasmid by disarming it, or we can say by modify it with some changes. This is how we can do that disarming TI plasmid, in which oncogenes are deleted from the tDNA and new gene of interest can be added to make the desired tDNA. It means we will delete auxin, cytokine, and opine region, because this is the responsible for the tumorigenesis, like tumor formation in the plant. So we don't need that. We don't need the auxin, cytokine, and opine region, because we want to insert our gene of interest, or we want to insert the desired gene. So we delete auxin, cytokine, and opine region. We cut that. This is how the size of TI plasmid somewhat decreases, and then we can join new gene of interest, which we want to transfer it to the plant, or host chromosome. So we use the naturally transfer mechanism of TI plasmid for uh, better use, or we can say for plant, mostly the plant benefits. We can use that. TI plasmid disarming can be done by, like, opine are not going to produce an tumorigenesis function delete. So these few things can be done, and we can select the transformed and non-transformed one by using two antibiotics, which in which one antibiotic is harmful for the bacteria, which can affect the agrobacterium tumefaciens cells, and the uh, next antibiotic is helpful for the selection of transformed plant cells and non-transformed plant cells. This is how we can select. That's the different process. It's basically disarming TI plasmid is beneficial because we delete the tumorigenesis part or tumorigenesis genes and replace it with the gene of interest, and then transferred into the plant cell for the beneficial functions. The next and the last topic for our presentation is RI plasmid. It's way similar to the TI plasmids as well. Just the difference is that the virulence plasmid of Agrobacterium rhizogenes. TI plasmid is of Agrobacterium tumefaciens. So RI plasmid means root inducing plasmid, and TI plasmid was tumor inducing plasmid. In RI plasmid cause formation of hairy root disease, like I have shown in the diagram that this kind of root formation occur. The root multiplication starts due to the RI plasmids, and it's similar with the TI plasmid. It also contains left border and right border, and the tDNA region is also present in between, which are responsible for the hairy root disease cause. Uh, so we can say that it is also a gram negative bacteria. It's also a soil borne, and all strain, its specificity is that all strain of agrobacterium rhizogenes are known to produce agrosinopine, that is also a type of opine. So basically RI plasmids, the strains containing RI plasmids or can agrobacterium rhizogenes produce agrosinopines, which is a specificity of RI plasmid containing bacteria. Next, this diagram of plasmid is containing ORI regions, and the regions of different promoter terminator, which are responsible for the disease causing genes. Next is the advantages and disadvantages. So advantages of the plasmids, like agrobacterium plasmid, that is TI plasmid, is that it's a naturally occurring plasmid. As we till now we know that it's a naturally occurring plasmid, and that's why its efficiency is very high as compared to the artificial one. Its efficiency is very high. Screening process is easy because we can insert selectable markers for the selection process, and there are many multiple markers we can use. So that's why screening process is very easy in TI plasmids. Next advantages is the single plasmid or two plasmid we can use, like binary vectors we can use, and it ultimately it also increases the efficiency of the plasmid. Next, the disadvantages are less, like duration is lengthy as compared to other vectors. The duration is lengthy, And the next disadvantages is artificially designed for the vector function. Like we have to disarm the ti plasmid, and we want to do if ti plasmid as a vector, we cannot use it directly because it contains the tumor genes. So we have to delete that genes which are responsible for the tumor, and then we insert the gene of interest and all. It's all we have to do it artificially. That's why that's the disadvantage, but disadvantages are less, advantages are more. So we can use ti plasmid, or we can modify it to the we can modify ti plasmid to the useful vector for the beneficial functions for the plants and other eukaryotic cells. Thank you so much. Hope you like the presentation. Thank you.